My name is Humphrey Fonge, and I am a professor at Université Laval, so that's in Quebec City. So I'm the PI of the project. The project focuses on developing what we call radioimmunoconjugates, which are biologics that are radiolabeled with a certain therapeutic isotope. So my project is funded under the stream of enabling studies. And so basically, what we are going to be doing in this project is to develop what we call GMP formulations or good manufacturing practice formulations. And that's why it falls under the enabling studies, because these are classical studies that would enable investigators to be able to conduct phase one or get approval and then conduct phase one clinical trials. This project actually focuses on two diseases because the antigen that I'm targeting, which is connectin 4, is overexpressed in multiple epithelial cancers. But for our purpose, we've demonstrated in a lot of very extensive preclinical studies funded by CIHR and multiple other funding agencies that radiolabeled versions of this biologic, of this immunotherapeutic that we developed, really has huge therapeutic benefits in triple negative breast cancer and also in non-small cell lung cancer. So we've shown in multiple mouse models that this compound is very effective. And so that's the first phase of the work. Again, the deliverables of the project is what we call GMP material. So the compound itself will be GMP produced. And then the second deliverable is going to be that we would actually apply and hopefully obtain approval to conduct a phase one clinical trial. So we'll apply for that with Health Canada. But at least this project will take us to that level, which is a huge undertaking. There's so many small milestones and deliverables in between that. But the big picture, the overall goal is to be able to get us to a point where we are talking to Health Canada and obtaining approval for phase one trial. So our funding just kicked in. I think the project just started 1 week ago, I think April 1. But without the BioCanRex, we wouldn't have been able to get going, right? Because it takes several steps to make any biologic molecule. And so that funding really helps us get started. The project fits in from two fronts. So the first one is it's an immunotherapeutic in itself. It's a radiolabeled antibody, which is an immunotherapeutic. But one of the significant additional contributions of this particular molecule is going to be that it synergizes with immune checkpoint blockade. It's on the one hand, it's an immunotherapeutic, but it has potential applications with other oncoimmunology platforms that really can enhance the therapeutic benefit of the product. So the addition, like using this compound as in combination treatment with other immuno-oncology platforms that others in the network are using, really has potential for significant benefits. So it really works from two fronts. So I just got a Canada Research Chair Tier 1 in radiopharmacy and translational cancer theranostics, right? The translational cancer theranostics is what I really want to emphasize on here, because my research really focuses on really trying to quickly move promising compounds, promising immunotherapeutics into the clinic, right? And so that's why there's that portion of my research here. And so bringing some of the technologies that I've been working on in the lab for the last decade or more, it's really the most satisfying part of the work. So, we start developing a lot of compounds. It takes so many years. And then you get to a point where you really want to see if it works in patients. It is very satisfying. But even more so is because when I started working with patient advocates, when I even just come to my email and I read emails from patients that may have just listened to an interview from somewhere, and they will write to me and say, "Well, I really will be excited to be part of your trial." And so then having funding or being funded by Biocan Rex to really help me help patients is it's the most satisfying thing in my in my life. So my ultimate goal is to really move these compounds very quickly into patient care. Even if it would only cause remission or really significantly improve survival in one patient, just one, I will be very happy. So that's what drives me. Is like taking the stuff to patients. I think the technologies that I'm working on have the potential to make really significant difference in the solid tumor space. So this one is on triple negative breast and non-small cell lung, but I also have another one on small cell lung cancer. I have another one going on. I'm talking about translational projects, not preclinical projects. Another one going or coming up very soon, coming up on colon cancer. So my goal is to be able to bring these compounds very quickly into phase one, and hopefully one or more of them would eventually get to phase three or phase two, where you can actually really make some significant impact on patient care, right? So the goal is just to be able to move at least the needle and advance patient outcomes.